Good evening. Am I audible? Am I visible? Good evening, Mad Max. Good evening, Ravi Kumar. So, if you're joining the session for the first time, this is Dr Shanmugapriya. I'm professor and head of the Department of Biochemistry at Government Thoothukudi Medical College, and I'm your Unacademy educator for biochemistry. And today's session is entire biochemistry revision in 90 minutes. Yeah, so I will be trying to complete the entire biochemistry in 90 minutes, but that will be conducted as two parts. Part 1 will be today and part 2 will be on Tuesday. Okay, so together if you watch both these sessions, yeah, you can get the PDF from the community. If you follow me on Unacademy app, you will be able to get the PDF. I will share the PDF by tonight. You can get it, Mad Max. So that is the plan. And before we start the session, let me tell you a few facts about Unacademy plans. And the first fact is to remind you about the All India mock test that is conducted by Unacademy. And it is scheduled on February 26th. It is at 9 a.m. Hi, Meena Krishnan. It is at 9 a.m. and if you ask me why should I attempt this test, it is for two reasons. One is the analytics will tell you real time what your rank is. So you will know clearly what your All India rank is. That's point number 1. And point number 2 is after you attend the test, you'll also be getting a fully recorded solution. Yeah, so top educators will be giving you a key, and it's not a return key, it will be a videographed key that you will get, so you'll be able to understand it conceptually. Good evening, Surya. Good evening, Max. Okay. Good evening, Murli. So that is about the All India mock test. And apart from All India mock test, there is continuously tests that are happening. We will be conducting grant tests and subject-wise tests. So attend as many tests as possible. And there is a good news for those of you who are preparing for the NEET PG, which is on 21st May. We have a special batch which is NEET PG 2022 All Educator Revision Batch. And what is so special about it? This batch has been curated in such a way that the schedule will give you time for self-study. The schedule will give you time for concept building. So every day there will be 4-hour session, and the schedule will be provided to you all well ahead. So every subject there will be a 4-hour session, after which there will be a self-study period, self-study test. And following after completing every subject, you will also be given a subject-wise test, and then the explanation will also be provided. And along with that, there are also grant tests that you will be able to attend. There are 8 grant tests if you participate in this batch. Okay, going to follow only this video for biochemistry. Okay, okay, Vijay. This class is for FMG or NEET PG. This is for NEET PG, Mohammad Jafar. Okay. So for you to be able to attend this batch, you should either have a plus subscription or an iconic subscription. Iconic subscription, as you all know, will give you access not only to Unacademy platform, it also gives you an access to PrepLadder platform. And this is a subscription plan. For NEET PG subscription plan, 3 months would cost you 12,500. But if you use any of the educators code, you get a 10% off. My code is C Shanmugapriya. You can use any educator's code. My code is C Shanmugapriya. If you use that, you will be able to get it for 3 months at the rate of 11,250. And for iconic subscription, can first year students definitely, yes, Sudeep, you can definitely attend this. First year students can attend. Okay. And for iconic subscription, there is a bonanza offer. This is applicable till 16 February or for the first 3,000 subscribers. The actual Thanks, Ravi Kumar. So the actual cost for 6 months subscription was 40,000, and this bonanza price would be 36,000. And if you use any of the educators code, again you get 10% off. So the final price would be 32,400 for a 6-month subscription, which is iconic subscription, which gives you an access to both Unacademy and PrepLadder platform. Okay. This subscription will also enable you to participate in all these comprehensive batches which we have planned for both FMG and for NEET PG. For FMG, there is one batch which is comprehensive batch which started on 9th February. The duration is 4 months. And for NEET PG, we have simultaneously 3 batches happening. One is high yield, eagerly waiting for this. Yeah, it will definitely be useful. You will be able to get an overall idea of biochemistry. And most of the previous year questions will also be covered in this session. Okay. So NEET PG high yield MCQ marathon batch, and there is image based question batch, there is ultra fast revision batch. So duration of all these batches would be 1 month. And these are our toppers in the latest NEET PG September 2021, and these are our toppers in the FMG December 2021. So that's all about Unacademy plans. Let's start the session now. This is the first question to you all. I will be happy if you all participate actively by giving answers in the live chat. Okay. So glucose and fructose are. Can anybody give me the answer for this question? Glucose and fructose are they functional isomers? Are they optical isomers? Are they stereoisomers, epimers? Very good, Prathamesh. I am very sure you know the answer. So glucose and fructose, can I get entire course of biochemistry from Unacademy Plus for next? Yes, you can get it. Very good, all of you. All of you have given the right answer. It is a functional isomerism. Both glucose and fructose are functional isomers. So just have a look at these two structures. The good news is you don't have to memorize any of these structures. Just observe these structures. Do you see that both have 6 carbon atoms? Yeah, because both have 6 carbon atoms, you call them as hexoses. And do you see that both glucose and fructose have multiple hydroxyl groups? Yes or no? They have multiple hydroxyl groups. This is one property that will be common among any carbohydrate. So all carbohydrates will have multiple hydroxyl groups. And what is the difference that you observe between glucose and fructose? What is the functional group that is present in glucose, all of you? Can you all answer that? Hi SSDN. Very good, Surya. So the functional group that is present in glucose is an aldehyde group. So glucose is an aldose, whereas fructose, what is the second carbon atom in fructose? The second carbon atom in fructose is a ketone group. So fructose is a ketose, glucose is an aldose. So what is the carry home message here? Both glucose and fructose have the same molecular formula, which is C6H12O6. But they have different functional groups. One is an aldose, the other one is a ketose. So glucose and fructose are functional isomers. Is that clear? Yeah, good, Prathamesh. Now capital D glucose and capital L glucose are they functional isomers, optical isomers, enantiomers, epimers? What do you think is the right answer? You're right, Blooming Heart. Now give me the answer for this question. Ravi Kumar says it is enantiomers. Dr. DS says it's optical isomers. Blooming Heart, optical isomers. Yes, this is why I gave you this question. Do not get confused here. Capital D and capital L are not optical isomers. Optical isomers are small d, small l or plus and minus. Capital D, capital L are enantiomers. Do you all understand this? Capital D, capital L are enantiomers. So what are optical isomers, all of you? Optical isomerism is based on the way an isomer turns a plane polarized light. If an isomer turns a plane polarized light towards the right side, what will you call it as? Towards the right side, you will call it as dextrorotatory, small d or plus form. If an isomer turns it to the left side, what will you call it as? You will call it as levorotatory, small l or minus forms. So tell me, what are optical isomers, all of you? Optical isomers are small d, small l, plus or minus. They are not capital D and capital L. So what are capital D and capital L? They are enantiomers. So what are enantiomers? Enantio means mirror. Do you all understand this? Enantio means mirror. So what are enantiomers? Enantiomers are exact mirror images. That is, you will have to change the orientation with respect to all the carbon atoms. Yeah, this is glucose. For you to call something as an enantiomer of glucose, you should change the orientation with respect to all the carbon atoms. But finally, you name them according to the orientation in the penultimate carbon atom, as Dr. Surya has mentioned. Is screen blurred? Yeah, so how do you name them? You name them according to the orientation in the penultimate carbon atom. What do you mean by penultimate? Ultimate is the last carbon atom. Penultimate is the 5th carbon atom. So in the 5th carbon atom, if OH lies on the right side, in the 5th carbon atom, if OH lies on the right side, that is called as capital D form. And in the penultimate carbon atom, if OH lies on the left side, that is called as capital L form. So can you see that here? Yeah, this is glucose and this is the first carbon atom, second, third, fourth, fifth. In the fifth carbon atom, if OH lies on the right side, what will you call it as? You will call it as capital D. And in the fifth carbon atom, if OH lies on the left side, that is called as capital L. So capital D and capital L are enantiomers. Don't get confused between enantiomers and optical isomers. Is that clear? So what is answer, all of you? Capital D and capital L are enantiomers. Okay. Now the next question. Alpha glucose and beta glucose are what? Alpha and beta glucose are what? Can anybody give me the right answer? Alpha and beta. Are they functional isomers? No, right? Optical isomers? No. Enantiomers? No. Avantika says it's C. Avantika, just now I said enantiomers are capital D and capital L. Okay. Blooming Heart says D. Very good. Rajaram says D. Yes. Answer is anomers. How do you remember it? Anomers starts with which letter? A. And it is alpha and beta. Alpha also starts with A. So anomerism is nothing but alpha and beta forms. Do you all understand this? Anomerism is nothing but alpha and beta forms. So let me tell you, ring forms. So let me tell you about types of isomerism. Isomerism is of three types. First tell me what is isomerism. Here the two molecules have the same molecular formula. Yeah, they have the same molecular formula, but they have either different structural formula or they differ in spatial orientation of their groups. So can you tell me what is isomerism? Isomerism is a property by which two or more molecules have the same molecular formula, the C6H12O6. Yeah, that will be the same. But what will they differ in? They either differ in structural formula or they differ in spatial orientation of their groups. And this isomerism is of three types. What are the three types, all of you? Optical isomerism, functional isomerism and stereoisomerism. So I want you to tell me what is optical isomerism. Can you all tell me what is optical isomerism? It is based on how they turn the plane polarized light. So what are the two types of optical isomers that you know? Small d, small l, or plus and minus. Okay. Now what are functional isomers? In functional isomers, they have the same molecular formula, but they have different functional groups. Yeah, and in carbohydrate, for every molecular formula, there will be a set of aldoses, there will be a set of ketoses. So aldoses and ketoses are functional isomers of each other, provided they share the same molecular formula. And to be very specific, for a molecular formula of C6H12O6, which is the well-known aldose, all of you, for a molecular formula of C6H12O6, which is the well-known aldose, the well-known aldose is glucose and the well-known ketose is fructose. So glucose and fructose are functional isomers. So are you clear about this so far? Yes. Now about stereoisomerism. In stereoisomerism, the two molecules will have the same molecular formula and the same structural formula. Do you understand this? In stereoisomerism, the two molecules will have the same molecular formula and the same structural formula, but they differ in spatial orientation of their groups. They differ only in spatial orientation of their groups. For example, you all know this is the structure of glucose, right? What happened? Yeah, you all know this is the structure of glucose. So in structure of glucose, if you maintain everything, you maintain everything, and if you change the orientation of H and OH groups with respect to the second carbon atom alone, and if you put OH here and H here, that will be called as mannose. Yeah, that will be called as mannose. So tell me, what are glucose and mannose, all of you? Glucose and mannose have the same molecular formula. They have the same structural formula, but they differ only in spatial orientation of their groups. So glucose and mannose are examples of stereoisomerism. Okay. Now take glucose, you maintain everything, but you change the orientation only with respect to the fourth carbon atom. In the fourth carbon atom alone, you put OH here and H here. That will be called as galactose. So glucose and galactose are examples of what? Glucose and galactose are examples of stereoisomerism. So have you understood what is stereoisomerism? Yeah, what is stereoisomerism? In stereoisomerism, the two molecules will have the same molecular formula and the same structural formula, but they differ only in spatial orientation of their groups. An example is this. Can you see that, all of you? Whatever I told you, can you observe it here? Glucose and mannose have the same molecular formula and the same structural formula. The only difference is orientation in the second carbon atom. Similarly, if you maintain everything and if you change the orientation with respect to the fourth carbon atom, what will you call it as? You will call it as galactose. So glucose and galactose are also examples of stereoisomerism. Okay, and this stereoisomerism is dependent on the presence of an asymmetric carbon atom. Yeah, just 2 years back it was asked. Number of stereoisomers possible for glucose or number of stereoisomers possible for fructose. So it is based on the presence of asymmetric carbon atom. Tell me, what is an asymmetric carbon atom? Can anybody give me an answer? Asymmetric carbon atom is a carbon atom in which all the four valencies are satisfied by different groups. Do you all understand this? Asymmetric carbon atom is a carbon atom in which all the four valencies are satisfied by different groups. Now look at the first carbon atom and tell me if it's asymmetric or symmetric. Yeah, the first carbon atom, is it asymmetric or symmetric? Do you see that two of its valencies are satisfied by O? Yeah, that makes it symmetric. Do you all understand this? That makes it symmetric. Now consider the last carbon atom. The last carbon atom, again, two of its valencies are satisfied by the same hydrogen atom. So that makes the last carbon atom also symmetric. Whereas consider the second carbon atom. In the second carbon atom, one valency is satisfied by CHO, second valency by H, third valency by OH, and fourth by this whole group. Right? That makes second carbon atom of glucose asymmetric. So in glucose, can you tell me how many asymmetric carbon atoms do you find? In glucose, excluding the first and the last, excluding the first and the last, the remaining four are asymmetric carbon atoms. How about fructose, all of you? In fructose, both the first carbon atom and the last carbon atom are CH2OH, and that makes them symmetric. Similarly, the functional carbon atom, that is also symmetric because it is C double bond O. So in fructose, excluding the first, last and the functional carbon atom, the remaining three are asymmetric. Yeah, the remaining three are asymmetric. So tell me, how many stereoisomers are possible in glucose? Number of stereoisomers possible in a molecule with n number of asymmetric carbon atom is 2 to the power of n. Yeah, number of stereoisomers possible in a molecule with n number of asymmetric carbon atoms is 2 to the power of n. So can you all tell me how many stereoisomers are possible for glucose? Yeah, 2 to the power of 4 is 16. So how many stereoisomers are possible for glucose? It is 16. How many in fructose? 2 to the power of 3 is 8. Yeah, 2 to the power of 3 is 8. So this is an introduction to stereoisomerism. Now what are the types of stereoisomerism? There are 4 types of stereoisomerism. The first one is anemomerism. You start with the first alphabet. What is it? It is anemomerism. The second one is epimerism. Yeah, the third one is Diastereo Isomerism, and the last one is Enantiomerism. So all of you tell me, what are the 4 types of stereoisomerism? The 4 types are anemomerism, epimerism, Diastereo Isomerism and Enantiomerism. Okay. So anemomerism is with respect to the anomeric carbon atom or the functional carbon atom. Do you all understand this? Anemomerism is with respect to the anomeric carbon atom or the functional carbon atom. In that case, it is with respect to which carbon atom in the case of glucose? Yeah, it is with respect to which carbon atom in the case of glucose? Can you all answer this? I am checking the live chat. Can you all please answer? In the case of glucose, it is with respect to the first carbon atom, and in the case of fructose, it is with respect to the second carbon atom. Very good. So in the case of glucose, it is with respect to first carbon atom. In the case of fructose, it is with respect to the second carbon atom. So in the functional carbon atom, listen to this carefully. In the functional carbon atom, if OH lies above the plane of the ring, that is called as beta form. Yeah, in the functional carbon atom, if OH lies above the plane of the ring, that is called as beta form. If OH lies below the plane of the ring, that is called as alpha form. So can you see that here, all of you? This is the functional carbon atom. In the functional carbon atom, if OH lies above the plane of the ring, what do you call it as? Above is beta. Yeah, if OH lies below the plane of the ring, that is called as alpha form. So tell me, all of you, alpha and beta, they are examples of what? Alpha and beta are examples of anemomerism. It is with respect to the functional carbon atom. Okay? Okay, bird fly up. Yeah, good one. Okay? So that is anemomerism. Now about epimerism. Listen to this carefully. In epimerism, the two molecules will have the same molecular formula and the same structural formula, but they differ in spatial orientation with respect to one of the asymmetric carbon atoms. They differ with respect to one of the asymmetric carbon atoms other than the penultimate carbon atom. So have a look at the structure of glucose, all of you. In structure of glucose, for you to call something as an epimer of glucose, that structure should differ from glucose with respect to one of the asymmetric carbon atoms. So what are the carbon atoms you will have to exclude? You have to exclude all symmetric carbon atoms. So I am going to exclude the first one and the last one. Yeah. So it has to differ with respect to one of the asymmetric carbon atoms other than the penultimate carbon atom. So I have excluded first, fifth and sixth. So what am I left with? Can you all answer this? What am I left with? I am left with the second or the third or the fourth carbon atom. So for you to call anything as an epimer of glucose, that structure should differ from glucose with respect to either second or third or fourth carbon atom. Okay, and don't we know already examples for this? Very good, blooming heart. Don't we know already examples for this? In the second carbon atom, if you change the orientation, what is it called as? Good afternoon, Suman. In the second carbon atom, if you change the orientation, what is it called as? Mannose. In the fourth carbon atom, if you change the orientation, what is it called as? Galactose. So to begin with, I just told you glucose and mannose are examples of stereoisomerism. Now, which type of stereoisomerism do they belong to? Glucose and mannose belong to epimerism. Similarly, glucose and galactose, previously I told you, they are examples of stereoisomerism. Which type of stereoisomerism do they belong to? They are epimers. Yeah, so glucose and mannose are epimers, glucose and galactose are also epimers. So that is all about epimerism. So what is epimerism? They should differ with respect to only one of the asymmetric carbon atoms other than the penultimate carbon atom. Now what is diastereo isomerism? There is only a minimal change or difference between epimerism and diastereo isomerism. So listen to this carefully. In diastereo isomerism, the two structures will differ with respect to one or more of the asymmetric carbon atoms. Do you understand this? They differ with respect to one or more of the asymmetric carbon atoms other than the penultimate carbon atom. Do you understand the difference now, all of you? Do you understand the difference between epimerism and diastereo isomerism? What is the difference? Can you all type it in the live chat? Epimers differ with respect to only one. Diastereo isomers differ with respect to one or more. Yeah. Example is, I will give you, how do you get mannose from glucose? You get mannose from glucose by changing the orientation in the second carbon atom. How do you get galactose from glucose? You get galactose from glucose by changing the orientation in the fourth carbon atom. Now I request you all to compare mannose and galactose. Are you all comparing? Yeah, can you compare mannose and galactose? Do you see that both mannose and galactose differ with respect to both second and fourth carbon atom? Yeah, they differ with respect to both second and fourth carbon atom. Very good, Saima. You are right. I will show you that. Yes. So mannose and galactose. Can you call mannose and galactose as epimers? You cannot call them as epimers because they differ with respect to more than one. So what should you call them as? You should call them as diastereo isomers. Okay, so diastereo isomers differ with respect to one or more of the asymmetric carbon atoms. So let me draw you this. Yeah, listen to this, watch this carefully. Yeah, epimers are subset of diastereo isomers. Do you understand this? Epimers are subset of diastereo isomers, because what are diastereo isomers? They differ with respect to one or more. Epimers differ with respect to only one. So all epimers are diastereo isomers. Do you all understand this? All epimers are diastereo isomers. Okay, so that's all about diastereo isomerism. Now what is enantiomerism? I want you to answer this. Haven't I told you about enantiomerism? What is enantiomerism? Enantio means mirror. So enantiomers are exact mirror images, right? They are exact mirror images, and then you name them according to the orientation in the penultimate carbon atom. In the penultimate carbon atom, if OH lies on the right side, what will you call it as? Capital D form. If OH lies on the left side, you will call it as capital L form. So capital D and capital L are enantiomers. And the other name for enantiomerism is racemism. Yeah, the other name for enantiomerism is racemism. So can you tell me what is a racemase enzyme? Can anybody tell me what is a racemase enzyme? Racemase is an enzyme which can interconvert capital D and capital L forms. Yeah, racemase is an enzyme which can interconvert capital D and capital L forms. Okay. Okay. So that's all about isomerism. So can we summarize isomerism by me asking you few questions and you giving me answers? Can you try doing that? Yeah. So glucose and fructose are examples of what? Glucose and fructose are examples of functional isomerism, right? Glucose and fructose are examples of functional isomerism. And then small d, small l, very good Blooming Heart, very good Surya, Aruna. Small d, small l, what are small d, small l? They are optical isomers. Okay, whereas what are capital D and capital L? Capital D and capital L are enantiomers. What is the other name for enantiomerism? It is racemism. Very good all of you. Kanjan, Saima, good. So capital D, capital L are enantiomers. The other name for enantiomerism is racemism. What are alpha and beta, all of you? Alpha and beta starts with which letter? A, and it's an example of anomerism. And what is alpha? In the functional carbon atom, OH lying below the plane of the ring is alpha. OH lying above the plane of the ring is beta. So alpha and beta are anomers. Okay. Glucose and mannose are examples of what? Glucose and mannose are examples of epimerism. At which position? Second position. How do we remember it? How many N's do you find in mannose? There are 2 N's in mannose. So glucose and mannose are epimers at which position? Second position. And glucose and galactose. Yeah, glucose and galactose are also examples of what? Epimerism. At which position? Fourth position. Okay. And what are the examples of diastereo isomerism? I told you mannose and galactose are diastereo isomers. Okay. So I hope it's clear now. Alpha and beta glucose are examples of anomerism. All of the following are reducing sugars except. Can you give me an answer for this? All of the following are reducing sugars except glucose, fructose, sucrose, maltose. So for this you will have to know when will you call. Very good Blooming Heart, very good Aruna Devi. Yes, it is sucrose. So for answering this Dawood, it is not glucose. Glucose is a reducing sugar. Okay, it is not fructose. Fructose is a reducing sugar. So for you to be able to answer this question, you need to know what is the criteria that needs to be satisfied by a carbohydrate for you to call it as a reducing sugar. Only those carbohydrates which have a free carbonyl group will be reducing. It's very simple. Those carbohydrates which have a free carbonyl group will be reducing. And that is why glucose, which has got a free carbonyl group in the first carbon atom, and fructose, which has got a free carbonyl group in the second carbon atom, are reducing sugars. Whereas how about sucrose, all of you? Yes, free functional group or free carbonyl group. Okay. How about sucrose? Do you all know that sucrose is a disaccharide? Yes or no? Sucrose is a disaccharide which is made up of glucose and fructose. So my question to you is, when you know sucrose is made up of two reducing sugars, why is sucrose non-reducing? Can you answer this question? When you know sucrose is made up of two reducing sugars, why is sucrose non-reducing? It is because you get sucrose by linking the first carbon atom of glucose. It is because you get sucrose by linking the first carbon atom of glucose with the second carbon atom of fructose. Do you see that here? Yeah, you get it by linking the first carbon atom of glucose with the second carbon atom of fructose. That way neither the carbonyl group of glucose nor that of fructose is free. Or in other words, if you are asked why is sucrose non-reducing, your answer should be because the linkage that is present in sucrose is alpha 1,2. Yeah, the linkage that is present in sucrose is alpha 1,2. Have you understood this now? Very good, Hanuman. Yeah, so sucrose is a non-reducing sugar. Okay, glucose and fructose are reducing. Maltose is also reducing. Why do we say maltose is reducing? Please say linkages of disaccharides. So how many of you want me to say about linkages of disaccharides? I can tell you now. Okay, I will tell you about linkages of disaccharides. There are 5 disaccharides about which you should learn always. Yeah. So what are the 5 disaccharides you know? The 5 disaccharides include maltose. Yeah. Maltose, isomaltose and trihallose. Yeah. Maltose, isomaltose, trihallose, and then you have sucrose. And the last one is lactose. Okay, Nairobi. Yes, I'm telling you now. So maltose, maltose, isomaltose, trihallose. The 4th one is sucrose. The 5th one is lactose. So first let me tell you what are the sugars that are present. Then we will discuss what are the linkages. So tell me what are the sugars which are present in maltose. Whenever you see the term malt, you can close your eyes and say it will have 2 glucose residues. Do you understand this? What does malt mean? Malt means multiple glucose residues. So maltose being a disaccharide, what do you think it will be made up of? It will be made up of 2 glucose residues. And what is isomaltose made up of? Again malt. What will malt be made up of? 2 glucose residues. Trihallose is also made up of 2 glucose residues. So tell me what are the 3 disaccharides which have 2 glucose residues? Maltose, isomaltose and trihallose. And they differ only with respect to the linkage that is present. Okay. Now what is sucrose made up of, all of you? That everybody knows, right? Sucrose is nothing but the table sugar which we take. And the sucrose is nothing but glucose and fructose. Just now I told you it is glucose and fructose. The last one is lactose. What is lactose made up of? Lactose has got galactose and glucose. Do not even change the sequence. For you to call something as lactose, it has to be first galactose and then it has to be glucose. That's why it's called as lactose. Okay. So that is about the individual sugars which are present in a disaccharide. Now about linkages. Yeah, now about linkages. I want you to know that carbohydrates prefer to get linked with alpha 1,4 linkages mostly. Do you all understand this? Most of the carbohydrates prefer to have what linkages along straight chains? Most of the carbohydrates prefer to have alpha 1,4 linkages along straight chains, and they prefer to have alpha 1,6 linkages at branch points alone. Do you know that we have branched carbohydrates? Yeah, starch is branched, glycogen is branched. So at the branch point, wherein branch point begins alone, you will find alpha 1,6 linkage. So have you memorized it? Yeah, along the straight chain, what is the linkage you will find? Along the straight chain, it is alpha 1,4 linkage. At branch point alone, it will be alpha 1,6 linkage. Now, what are the exceptions that we have to learn about? The first set of exception is related to maltose, isomaltose, trihallose. And if I tell you maltose follows the thumb rule. Yeah, maltose follows the thumb rule. So tell me what is the linkage that is present in maltose? If maltose follows the thumb rule, very good. The linkage that is present is alpha 1,4. Isomaltose has got alpha 1,6. Yeah, S for 6. Isomaltose has got alpha 1,6. Trihallose has got alpha 1,1. Okay, trihallose has got alpha 1,1 linkage. Now sucrose, I will wait for you to answer. Can you tell me what is the linkage that is present in sucrose? Yes, very good Nairobi, good correlation. Yes, debranching enzyme defect is alpha 1,6 breaking point. Sucrose, why is sucrose non-reducing? I told you sucrose is non-reducing because the linkage that is present is alpha 1,2. It is because you get it by linking the first carbon atom of glucose with the second carbon atom of fructose. Yeah. So the linkage that is present in sucrose is alpha 1,2. And lactose. Yeah, listen to this carefully. Most of the human digestive enzymes cannot attack beta linkages. Do you know that? Most of the human digestive enzymes cannot attack beta linkages, and that is why we cannot digest cellulose. Don't you know that plant diet provides us with fiber? Yeah, you know plant diet provides us with fiber. When I say it provides us with fiber, it means we cannot digest it. Why do you think we cannot digest it? Because plant cells have, plant cell wall has cellulose. Okay, and this cellulose has got what linkage? Cellulose has got beta linkages. Cellulose has got beta 1,4 linkage. So if you are asked about cellulose, what should be your answer? Your answer should be cellulose is made up of multiple glucose residues that are linked by what linkages, because of which we cannot digest it? It is beta 1,4 linkage. And if I tell you that the only human digestive enzyme which can attack beta linkage is lactase. Yeah, the only human digestive enzyme which can attack beta linkage is lactase. What does that tell you about lactose? What does that tell you about lactose? Yeah, lactose has got beta 1,4. Yeah, lactose has got beta 1,4 linkage. So are you clear about these linkages? Yeah. So can I ask you, will you be able to answer this? Nairobi, yes, Nairobi. Whenever we talk about linkages, we denote only the first anomer form. Suppose maltose has got 2 glucose residues. And we check what is the anomer form in which the first sugar exists. What is the first sugar here? It is glucose. Glucose here is in alpha form. So we call it as alpha 1,4 linkage. Do you understand that, Nairobi? We are not bothered about what the second sugar is in, because the enzyme has to attack only that linkage, right? Yeah. So we don't denote what the second sugar anomeric form is. We always denote the first sugar anomeric form. So we generally write it as alpha 1,2. Okay. So tell me, let's start with the first sugar, maltose. Quickly tell me, what is maltose made up of? Maltose, isomaltose, trihallose, all three are made up of what? All three are made up of 2 glucose residues. They differ only with respect to the linkage that is present. And the fourth disaccharide is sucrose. What is sucrose made up of? Sucrose is made up of glucose and fructose. Very good. And what is lactose made up of? Lactose is made up of galactose and glucose. Now tell me the linkage. What did I tell you about maltose, all of you? Maltose follows the thumb rule. So what is the linkage present in maltose? Alpha 1,4. Isomaltose, S for 6. So isomaltose has got alpha 1,1. Trihallose, tell me what is the linkage present in trihallose? Trihallose is isomaltose alpha 1,6. Trihallose is alpha 1,1. Okay, trihallose is alpha 1,1. Sucrose. Why is sucrose non-reducing, all of you? Sucrose is non-reducing because the linkage that is present is alpha 1,2. And then lactose. What is the clue that I gave you to remember this? Lactase is the only human digestive enzyme which can attack beta linkage. Yeah, so lactase, beta linkage is attacked. So lactose has got beta 1,4 linkage. Okay, lactose has got beta 1,4 linkage. Okay. So can you tell me what are the non-reducing disaccharides? Can you all tell me what are the non-reducing disaccharides? One is sucrose, right? Sucrose is non-reducing. The other one is trihallose. Non-reducing disaccharides are number 1 sucrose, the other one is trehalose. So why is trehalose also non-reducing? Hi Meena, tell me, you have something to say? Why is trehalose also non-reducing? Because what are the sugars present in trehalose? The sugars present in trehalose are 2 glucose residues. And in both the glucose residues, where do you have the free carbonyl group? In the first carbon atom. And what is the linkage that is formed? It is alpha 1,1. So neither the carbonyl group of first glucose nor that of second glucose is free. So all of you tell me, what are the 2 non-reducing disaccharides? The 2 non-reducing disaccharides are sucrose and trehalose. Okay. The test that is used to detect a reducing substance in the given solution is. Can you all answer this? The test that is used to detect a reducing substance in the given solution is. I hope you all know the answer. Yeah, it is Benedict's test. Very good, Surya. It is Benedict's test. So, Benedict's test is a test that is done to detect reducing sugars. So, what is the use of Biuret test, all of you? Biuret test is used to identify proteins. Yeah, it is used to identify proteins. And what is the purpose of Molish test? Molish test is a group test done for carbohydrates. Yeah, Molish is for carbohydrates. Benzidine is a test that is done to detect blood. Yeah, benzidine is a test that is done to detect blood. So tell me, Benedict's is for reducing sugars. Molish test is for all carbohydrates. Molish is for all carbohydrates. Biuret test is for proteins. Benzidine test is for blood. Okay. So can we learn about few color reactions, few tests that can be asked in the exam? Yeah. Abnormal constituents, the tests. Yeah. So the first one is, one second. Yeah. So the first one is reducing sugar. Can you all answer me? Reducing sugar is detected by which test? It is Benedict's test. And what is the result that you observe? If it is positive, the blue color Benedict solution will turn into green, or it becomes yellow, or it becomes orange or red. If it is positive, the blue color Benedict solution will turn slowly into green or yellow or orange or red, and the color will depend upon the concentration of the reducing substance that is present in the given solution. So that's about reducing sugars. The next one is ketone bodies. This has been repeatedly asked. They ask you this as an image based question or as a case based question. So ketone bodies, what is the test that is done? It is Rothera's test. And what will be the image that is generally given? The image, there will be a purple ring. Yeah, if you see a purple ring, what should be the clue? Yeah, purple ring is a ring that is found in Rothera's test. Rothera's test is a test that is done to detect ketone bodies. Okay. And then how about proteins? What is a test that we do to detect proteins? Just now I said Biuret test, right? But Biuret test will not be able to detect protein in urine because urinary protein concentration is very low. And Biuret test is not that sensitive to detect protein in urine. So if it is protein in urine, it is sulfosalicylic acid test. Yeah, protein in urine is sulfosalicylic acid. Xanthoprotic acid is a test that is done to detect aromatic amino acids, Joy. Yeah, xanthoprotic acid test is a test that is done to detect aromatic amino acids, not for proteins. Aromatic amino acids including phenylalanine, tyrosine and tryptophan. Okay. For protein in urine, what is the test that is done? It is sulfosalicylic acid test, which gives you white precipitate. You don't have to write anything. I will be providing the filled tabular column as PDF. Yeah. I will be sharing it in the community tonight. Okay. So protein sulfosalicylic acid test, it is white precipitate. And then blood. What is the test that is done to detect blood, all of you? It is benzidine test. Yeah, for blood it is benzidine test, where you will find greenish or black precipitate. It will form a green or black precipitate if the blood is present. Actually, it is a test that is done to detect heme. Okay, heme will answer by giving green or black precipitate. And then what is the test that is done for bile salts? All of you? Bile salts is a favorite test of all undergraduate students. It is Hayes test. Do you remember you using sulfur powder, dropping it on the urine and then seeing whether it is floating or is it sinking? Yeah, that is Hayes test. That is a test that is done to detect bile salts. And what will you find here? In this case, you are going to find that sulfur sinks to the bottom. If sulfur sinks to the bottom, then there is bile salts. Okay. And then bile pigments. The test done to detect bile pigments is Fauchett's test. Bile pigment is Fauchett's test. So how do you perform Fauchett's test? Do you remember that you get a precipitate and then you filter it? The filter paper will have the precipitate, and to that precipitate you add ferric chloride solution. If there is greenish discoloration, if there is greenish discoloration of the precipitate that is found in the filter paper, then that is positive. Okay, so these are the color reactions. Benzidine test, blood, benzene, blood cancer. Yeah, good correlation. Yes. Okay, so these are the tests that can be done to detect abnormal constituents in urine. So what are the abnormal constituents I've told you about? Starting from reducing sugar. Can you all answer what is the test done to detect reducing sugar? Reducing sugar is Benedict's test, wherein blue color becomes green or yellow or orange or red. Okay, next ketone bodies. What is the test for ketone bodies? It is Rothera's test, when you will find a purple colored ring. And then we have proteins. Proteins in urine can be detected by sulfosalicylic acid test. White precipitate is formed. And then blood or heme in urine can be detected using benzidine test. Okay. And then bile salt, Hayes test, wherein the sulfur powder sinks to the bottom. For bile pigments, what is the test? It is Fauchett's test. Yeah, Fauchett's test, what will you see? You will see greenish discoloration in the filter paper after you add ferric chloride. Okay. So that is about the tests. Okay. So the test that is used to detect a reducing substance in the given solution is Benedict's test. The rate limiting enzyme of glycolysis is? Can you all answer this? The rate limiting enzyme of glycolysis is? It's a very simple question but frequently asked. It is phosphofructokinase 1. Yeah, it is phosphofructokinase 1, which is the rate limiting enzyme of glycolysis. Very good, Dr. DS. Very good, Armugam. Nairobi, it is not fructose 1,6-bisphosphatase. Fructose 1,6-bisphosphatase is the rate limiting enzyme of gluconeogenesis. So can we learn about rate limiting enzymes of carbohydrate metabolism? Yeah, let's quickly revise it. So the pathway glycolysis. Tell me what is the rate limiting enzyme of glycolysis? It is phosphofructokinase 1. Okay. And then what is the rate limiting enzyme of citric acid cycle, TCA cycle? For citric acid cycle, it is isocitrate dehydrogenase. For citric acid cycle, the rate limiting enzyme is isocitrate dehydrogenase. And then glycogen synthesis. What is the rate limiting enzyme of glycogen synthesis? It is glycogen synthase. Obviously, right? For glycogen synthesis, it is glycogen synthase. For glycogenolysis, yeah, for glycogenolysis, it is glycogen phosphorylase. For glycogenolysis, it is glycogen phosphorylase. And then gluconeogenesis. What is the rate limiting enzyme of gluconeogenesis? The rate limiting enzyme of glycolysis is phosphofructokinase 1. So the enzyme which reverses phosphofructokinase 1 should be the rate limiting enzyme of gluconeogenesis. So what is the rate limiting enzyme here? It is fructose 1,6-bisphosphatase. For gluconeogenesis, it is fructose 1,6-bisphosphatase. And finally, HMP shunt. So the rate limiting enzyme of HMP shunt is glucose 6-phosphate dehydrogenase. Yeah, it is glucose 6-phosphate dehydrogenase. So tell me what are the rate limiting enzymes I've told you about. You should not commit a mistake on this. Yeah. So glycolysis, what is the rate limiting enzyme? It is phosphofructokinase 1. For citric acid cycle, it is isocitrate dehydrogenase. For glycogen synthesis, it is glycogen synthase. For glycogenolysis, it is glycogen phosphorylase. For gluconeogenesis, it is fructose 1,6-bisphosphatase. And for HMP shunt, it is glucose 6-phosphate dehydrogenase. Okay. So what is the answer, all of you? Very good. What is the answer? The rate limiting enzyme of glycolysis is phosphofructokinase 1. Products of anaerobic glycolysis is. Can you give me the right answer for this question? Products of anaerobic glycolysis is? Is it pyruvate 7 ATP, lactate 32 ATPs? Very good, Surya. Very good, Meda. So products of anaerobic glycolysis will be 2 lactate and 2 ATPs. Yeah, 2 lactate and 2 ATPs. So let me tell you few facts about glycolysis which can be asked as MCQs. Okay. So what is glycolysis? Glycolysis is a process by which you split glucose. Yeah, it's a process by which you split glucose to form 2 products. And the 2 products which you get will differ depending upon whether the process occurs aerobically or anaerobically. What are the products that you get from aerobic glycolysis, all of you? Aerobic glycolysis will give you 2 molecules of pyruvate and 7 ATPs. Whenever I tell you a number of ATP, memorize it immediately. Now we won't be able to explain how we get 7 ATP. That's not, this is not the time. So aerobic glycolysis will give you how many ATPs? It will give you 2 pyruvate and 7 ATPs. Anaerobic glycolysis gives you 2 lactate and 2 ATPs. Anaerobic glycolysis will give you 2 lactate and 2 ATPs. So can you tell me what is the significance of glycolysis? Based on this, can you tell me what is the significance of glycolysis? Glycol is the only pathway that can generate ATP even in the absence of oxygen. Do you understand this? Glycol is the only pathway that can generate ATP even in the absence of oxygen. So why do you want a pathway to generate ATP in the absence of oxygen when you know we are all aerobic organisms? Though we are all aerobic organisms, we have some cells like RBCs, retinal cells and corneal cells. So what is so special about RBCs, retinal cells and corneal cells? Dr. DS says energy production. Dr. DS, there are so many pathways in our body which can provide more number of ATPs than what glycolysis can. Okay, so that is not the significance of glycolysis. When I ask you for a significance, it's something which is unique about the pathway. So what is unique about the pathway? It can generate ATP in the absence of oxygen. So we have some cells like RBCs, retinal cells and corneal cells which do not have mitochondria. So they are totally dependent on glycolysis for ATP production. Okay. Similarly, white muscle fibers. Why are white muscle fibers white? Or why are red muscle fibers red? Red muscle fibers are red because they have myoglobin. And what is myoglobin, all of you? Myoglobin is the storage form of oxygen. Hemoglobin is the, hemoglobin is the transport form of oxygen. Myoglobin is the storage form of oxygen. So red muscle fiber with myoglobin, which is a storage form of oxygen, can undergo aerobic metabolism. But white muscle fibers without myoglobin, without a storage form of oxygen, will have to undergo what metabolism? Anaerobic metabolism. So white muscle fiber is dependent on what? It is dependent on anaerobic glycolysis. Okay. And then it is renal medulla. The third one is renal medulla. Do you all know that renal medulla receives a sparse blood supply? Yes or no? Renal medulla receives a very low blood supply to maintain the medullary interstitial hyperosmolality. So when the blood supply is low, it means they don't have a good access to oxygen. So they are also dependent on anaerobic glycolysis. So can you all tell me what are the cells which are totally dependent on glycolysis for ATP production? RBCs, retinal cells, corneal cells, they are totally dependent because they don't have mitochondria. White muscle fibers, because they don't have myoglobin, they are also dependent on glycolysis. And then renal medulla. Okay, so this should tell you about fuel utilization by all cells. Yeah, just based on this, you should be able, because in another 2 minutes, I will be telling you about the preferred fuel for every cell. Yeah, because that's being often asked, right? What is the preferred fuel for cardiac muscle fiber? What is the preferred fuel for liver when in the well-fed state? What is the preferred fuel for liver when the person is in starvation? Yeah. So for you to answer such questions, you need to know how do we choose a preferred fuel for a cell. That choice depends upon whether the cell is aerobic or anaerobic. Yeah, the choice depends upon whether the cell is aerobic or anaerobic. If it's an anaerobic cell, then no other choice. Yeah, what should it use? It should use only glucose as its fuel. Do you all understand this? If it's an anaerobic cell, no other go. It has to use only glucose as its fuel because only glucose can provide ATP anaerobically. But if it's aerobic cell, then the cell will have two choices. Yeah, aerobic cells will have two choices. The cell can either utilize glucose. Yes, RBCs, glucose, fed state and starvation. Very good. Be it fed state or starvation, RBCs use only glucose. So if it is aerobic cell, then the cell will have two choices. The cell can utilize either glucose aerobically or fatty acid aerobically. Given a choice, what will a cell choose? All of you listen to this and tell me. If it's aerobic cell, the cell has got two choices. It can use either glucose aerobically or fatty acid aerobically. Given a choice, what will a cell choose? What will a cell choose? Very good, Blooming Heart. The cell should choose fatty acids because fatty acids on oxidation will provide more number of ATPs than glucose on oxidation. Haven't you learned that palmitic acid on complete oxidation gives you 106 ATPs? Yeah, all of you memorize it. Even if you don't know this prior, memorize it now. Palmitic acid on complete oxidation gives you how many ATPs? 106 ATPs. Whereas glucose on oxidation gives you 32 ATPs. So if I give you a choice, what will you choose? You are going to choose only fatty acids. So all aerobic cells will utilize fatty acid as the preferred fuel. Okay, that is why what is the preferred fuel for cardiac muscle fibers, all of you? Cardiac muscle fibers are known for their mitochondrial content. So they are going to undergo aerobic metabolism. So what is the preferred fuel for cardiac muscle fiber? It is fatty acid. What is the preferred fuel for red muscle fibers, all of you? Red muscle fibers have myoglobin. So what metabolism will they undergo? Aerobic metabolism. If it is aerobic metabolism, what will be the preferred fuel for red muscle fiber? Again, fatty acid. So what will red muscle fiber store? Red muscle fibers store fatty acid as triacylglycerol and cholesterol ester. Right? Red muscle fiber will store fatty acid as triacylglycerol and cholesterol ester. And that is why you should refrain from having red meat. Have you heard of this? Yeah, generally we ask people to refrain from having red meat. Why? It is because if you consume red meat, then you will end up getting hypertriglyceridemia and hypercholesterolemia. Okay, so that's about preferred fuel for cardiac muscle fibers and red muscle fibers. But there is one exception to this rule. Though I said all aerobic cells prefer to utilize fatty acid, there is one exception. There is one aerobic cell which uses glucose, and that is neurons. Yeah, there is one aerobic cell which uses glucose, and that is neuron. Why? Why do you think neurons? Yes, creatine phosphate is also used by muscle during exercise. During exercise, the initial few seconds ATP will be provided by creatine phosphate. Okay, so tell me, why do neurons utilize glucose and they don't use fatty acid? Neurons do not use fatty acid because fatty acids in circulation are attached to albumin. And anything which is attached to albumin, can it cross the blood-brain barrier? Anything which is attached to albumin, can it cross blood-brain barrier? No. And that is why neurons, because they don't have an access to fatty acid, neurons have evolved in such a way that they preferably utilize glucose aerobically. Yeah, they preferably utilize glucose aerobically. So can we discuss the choices of fuels now? Yeah. Okay. Oh, I forgot to do it. Okay. 1 second. So I will now tell you about choices of fuels. The first is, so I will tell you what is a choice of fuel in the well-fed state and what is a choice of fuel in starvation. Okay. So the first cell that I am going to discuss about is RBCs. Can you all tell me What will be the preferred fuel for RBCs, be it well-fed state or starvation? I think already one other person gave the answer. I think it was Dr. somebody. Yeah, be it well-fed state or starvation, because RBCs do not have mitochondria. The only fuel RBCs can utilize is glucose. So RBCs are totally dependent on glucose through glycolysis. Okay, and neurons. Tell me about neurons. Though neurons are aerobic cells, what is their preferred fuel? They also prefer to utilize glucose in the well-fed state. In starvation also they prefer to utilize glucose, but they start utilizing ketone bodies. Ketone bodies can cross blood-brain barrier. So neurons in starvation continue to utilize glucose as a preferred fuel, but they start utilizing ketone bodies. Do you understand this? So that's about neurons. And then how about cardiac muscle fibers? All of you tell me about cardiac muscle fibers. Just now I told you cardiac muscle fibers are known for their mitochondrial content. They utilize aerobic metabolism. If it's aerobic metabolism, what will be their preferred fuel? Fatty acid. In the well-fed state and in starvation, they utilize fatty acid. But in starvation, they start utilizing ketone bodies. Okay. Now about skeletal muscle and adipose tissue. Yeah, skeletal muscle and adipose tissue. Why have I clubbed them both together? Can anybody tell me why have I clubbed them both together, skeletal muscle and adipose tissue? Do you all know that insulin dependent glucose uptake? Very good, Dawood. Do you know that insulin dependent glucose uptake happens in skeletal muscle, cardiac muscle and adipose tissue? Even if you don't know this prior, memorize it. It's a very essential fact that everybody should know. Yeah, insulin dependent glucose uptake happens in skeletal muscle, cardiac muscle and adipose tissue by GLUT4 transporter upregulation. Okay, in that case, tell me what will happen in the well-fed state. In the well-fed state, there will be insulin. Insulin causes upregulation of GLUT4 transporters in both skeletal muscle and adipose tissue. So what will they preferably utilize? They are going to utilize glucose. Do you all understand this? They are going to preferably utilize glucose. And what happens in starvation? In starvation, both will be utilizing fatty acids. Because in starvation, there will be peripheral lipolysis, right? There will be peripheral lipolysis and all fatty acids will be there in circulation. So both these cells can start utilizing fatty acid in starvation. So that's about skeletal muscle and adipose tissue. Now the last one is hepatocytes. So tell me what is the preferred fuel for hepatocytes in well-fed state? So long I have been telling you only glucose or fatty acid, right? Yeah, I have been only telling you glucose and fatty acid. Yeah, I will ask you questions after this. We will revise it. Yeah. So tell me about hepatocytes. What is the preferred fuel for hepatocytes in well-fed state? Interestingly, hepatocytes utilize amino acid in the well-fed state. Dr. DS says it's fatty acid. DS, it is not fatty acid because can you tell me how is fatty acid absorbed from the intestine? Yeah, can you all tell me how is fatty acid absorbed from the intestine? From the intestine, fatty acid is absorbed as chylomicron. Yes or no? It is absorbed as chylomicron. And does chylomicron get into blood vessel or does it get into lymphatics? Chylomicron, does it get into blood vessel or into lymphatics? Because the size of chylomicron is larger, it cannot be drained by capillaries. Because it is not drained by capillaries, it cannot get into portal vein. Do you understand this? Only when something gets into portal vein, it can reach the liver. Yeah, in this case, because fatty acids do not reach the liver. Why don't they reach the liver? Because fatty acids are absorbed in the form of chylomicron. Chylomicron gets into lymphatics. Lymphatics gets into thoracic duct. Thoracic duct gets into left subclavian vein, which gets into systemic circulation. Yeah, chylomicron does not get into portal circulation. Chylomicron gets into systemic circulation. Through systemic circulation, only extrahepatic tissues will get to meet your dietary lipid. Yeah, have you all understood this? Any dietary lipid which you all take, they do not reach the liver. Why don't they reach liver? Because they are absorbed as chylomicron. Through chylomicron, they get into systemic circulation. So only extrahepatic tissues get to meet the dietary lipid. So it is not going to be lipid. Do you all understand this? It's not lipid in the well-fed state. Now why not glucose? It is because liver has got glucokinase enzyme. Yeah, liver has got glucokinase enzyme which has got very low affinity for glucose. So that any glucose in the diet is used only by extrahepatic tissues. Okay, only when there is excess, liver will start using it as glycogen. So what is the preferred fuel, all of you? Liver in well-fed state, glucose, and starvation, amino acid, ma'am. That is wrong. Liver in well-fed state, it is amino acids. In the well-fed state, the protein in the diet. Yeah, only in the well-fed state, protein in the diet will become amino acids. That amino acids will reach liver and liver will be using it. What happens in starvation? In starvation, there will be peripheral lipolysis. And when there is peripheral lipolysis, adipose tissue triacyl glycerol will form fatty acid and all fatty acids reach liver. So what happens in starvation? In starvation, hepatocytes will start using fatty acids. In starvation, hepatocytes will start using fatty acids. So have you understood this? Yeah. So can we recollect this? Yes. So tell me, what is the preferred fuel? This recollection is for your sake. Please answer it along with me. Well-fed state and starvation. Yeah. Which cell did we begin with? We started with RBCs. So tell me, what is the preferred fuel for RBCs? Be it well-fed or starvation, it is always glucose. Well-fed or starvation is always glucose. And then how about neurons? Neurons also prefer to utilize glucose both in well-fed state and in starvation. But the difference is neurons will also start utilizing ketone bodies. Okay. How about cardiac muscle fibers? Okay, I should write it on the left side. Okay. So how about cardiac muscle fibers? Cardiac muscle fibers preferably utilize fatty acids both in well-fed state and in starvation. And again, cardiac muscle fibers start utilizing ketone bodies. Yeah, this is about cardiac muscle fibers. Skeletal muscle and adipose tissue. So why have we clubbed both skeletal muscle and adipose tissue together? It is because insulin dependent glucose uptake. All of you tell me, insulin dependent glucose uptake happens where? It happens in skeletal muscle, cardiac muscle and adipose tissue. By which transporter upregulation? By GLUT4 transporter upregulation. So in the well-fed state, for both skeletal muscle and adipose tissue, there is a good access to glucose. Okay. And what happens in starvation to skeletal muscle and adipose tissue? Fatty acids. Okay. And then the one which is totally different from all these cells is liver. So what should you remember about liver? You should remember that in well-fed state, lipids do not reach liver. Lipids go only to the extrahepatic tissues. Though carbohydrates reach liver through portal vein, liver has got an enzyme glucokinase, just got very low affinity. Okay, so it's not going to use glucose, it's not going to use fatty acid. What will it use? It is using amino acids. Very good. And what happens in starvation to liver? It is fatty acids. Okay, and liver cannot utilize ketone bodies. Do you all know that? If you are asked which organ cannot utilize ketone bodies, your answer should be liver. Because liver lacks which enzyme? Because liver lacks thiophorease. Okay. So quickly answer this question. We have already discussed about this. The cells which are dependent on glycolysis are all except. The cells which are dependent on glycolysis are all except. Are very good, Ashok. Excellent. Yes, Zakira. Excellent. So cells which are dependent on glycolysis are all except. RBCs are dependent. Yeah, white muscle fibers are dependent. Neurons are also dependent. Very good. The answer is cardiac muscle fibers, because what is the preferred fuel for cardiac muscle fibers? It is fatty acids. Which of the following is an irreversible step of glycolysis? I will wait for 30 seconds for you to try answering this. Which of the following is an irreversible step of glycolysis? Phosphoglycerate kinase, glyceraldehyde 3-phosphate dehydrogenase, pyruvate kinase and phosphotriose isomerase. What is answer? Very good, Staphylococcus. Yes, it is pyruvate kinase. Yeah, irreversible step of glycolysis is pyruvate kinase. So now I'm going to tell you the irreversible steps of glycolysis. And if you're asking me, why should we know the irreversible steps of glycolysis? Not just because it's asked in exam, but also for you to understand steps of gluconeogenesis. You know that, right? Gluconeogenesis is reversal of glycolysis. So to understand gluconeogenesis, you will have to know what are the irreversible steps of glycolysis, and you should know a means of reversing them. So what are the irreversible steps of glycolysis? The first step is irreversible, hexokinase or glucokinase. And then the rate limiting enzyme. What is the rate limiting enzyme of glycolysis? It is phosphofructokinase 1. And the last step, what is the last step enzyme? Pyruvate kinase. Okay, so tell me what are the 3 irreversible steps of glycolysis? All of you please memorize it, it's very essential. So what are the 3 irreversible steps of glycolysis? The first one, hexokinase or glucokinase, and then the rate limiting enzyme, which is phosphofructokinase 1, and then pyruvate kinase. Okay, so what does pyruvate kinase convert? Pyruvate kinase converts phosphoenol pyruvate to pyruvate. Yeah, do not just memorize the numbers. Yeah, because in the exam they will ask you only enzymes, they won't ask you numbers. Okay, so just memorize the enzymes which catalyze irreversible steps. So answer is pyruvate kinase. Okay, good, Staphylococcus. Which is an example of substrate level phosphorylation? Answer this, which is an example of substrate level phosphorylation? Acyl-CoA dehydrogenase, glyceraldehyde 3-phosphate dehydrogenase, succinate dehydrogenase, and phosphoglycerate kinase. Yeah, I will give you a clue. It is not A, it is not B. Yes, it is D. Okay, I will give you a clue. If you see the term dehydrogenase in an enzyme's name, yeah, if you see the term dehydrogenase in an enzyme's name, they are mostly an example of oxidative phosphorylation. Yeah, dehydrogenase enzymes are mostly examples of oxidative phosphorylation. Whereas if you see the name of the enzyme ending with kinase, they are mostly examples of substrate level phosphorylation. I am not saying always. Yeah, just a clue, just to exclude answers. Okay, so let me tell you what is substrate level phosphorylation, what is oxidative phosphorylation. When I tell you the difference, you will understand it better. So both substrate level phosphorylation, oxidative phosphorylation are types of catabolism. Yeah, both substrate level phosphorylation, oxidative phosphorylation are types of catabolism. All of you tell me what is catabolism. Simpler terms, can you tell me what is catabolism? Catabolism is a process by which we break down complex biomolecules into simpler substances, and that is done by breaking covalent linkages. Whenever you break covalent linkages, what is liberated? Energy is liberated, and depending upon how you trap the energy in the form of ATP. Yeah, when energy is getting liberated in catabolism, depending upon how you trap the energy in the form of ATP, catabolism is of 2 types. One is substrate level phosphorylation, the other one is oxidative phosphorylation. So what is substrate level phosphorylation? In substrate level phosphorylation, a set of substrates are getting converted to a set of products. Okay, and simultaneously you see that ADP is getting converted to ATP. Yeah, simultaneously you see that ADP is getting converted to ATP. In that case, you will call it as an example of substrate level phosphorylation. Do you understand the point here? It is so called because at the substrate level itself, you are trapping the energy in the form of ATP. That is why it is called as substrate level phosphorylation. To make it simple, in any chemical reaction, if you see substrates are becoming products, and simultaneously and directly if you see ATP is getting generated, that is an example of substrate level phosphorylation. Now what is oxidative phosphorylation? Even in oxidative phosphorylation, a set of substrates are getting converted to a set of products. Even here energy is getting liberated, but this energy is trapped in the form of not as ATP. The energy is not directly trapped as ATP. The energy is trapped as reducing equivalents like NADH or FADH2. Do you understand this? Here you are trapping the energy in the form of NADH or FADH2. Then you oxidize NADH and FADH2. Can you tell me what is oxidation, what is reduction in terms of electron transfer? What is oxidation, what is reduction in terms of electron transfer? The mnemonic that you can have is LEO. What is LEO? Loss of electron is oxidation. So gain of electron is reduction. Do you all understand this? Loss of electron is oxidation, gain of electron is reduction. So when I say I am oxidizing NADH and FADH2, I'm allowing them to leave electrons. Do you understand this? When I oxidize NADH and FADH2, they leave out electrons, and those electrons are transported in electron transport chain, which in a complex way helps in converting ADP to form ATP. Which in a complex way helps in converting ADP to form ATP. That is called as oxidative phosphorylation. Do you understand the difference? So tell me, what is substrate level phosphorylation? What is oxidative phosphorylation? In substrate level phosphorylation, directly you get ATP. In oxidative phosphorylation, you first get NADH or FADH2. Then you push NADH and FADH2 through electron transport chain, convert them into ATP. That is oxidative phosphorylation. And at this point, I want to tell you all one fact. It is never NADPH. Do you all understand this? It is never NADPH. NADPH can never get into electron transport chain. It is not going to give rise to ATP. And if it's NADH, as you have all answered it, NADH will give you 2.5 ATPs. FADH2 will give rise to 1.5 ATPs. Okay. Now between these two, which kind of phosphorylation do you think is more common? Do you think substrate level phosphorylation is common or oxidative phosphorylation is common? In human metabolism, because humans are aerobic organisms, between the two, which do you think is common? Between the two, oxidative phosphorylation is very common. Very good. Between the two, in human metabolism, oxidative phosphorylation is common, and there are only 4 examples of substrate level phosphorylation which I think you have to memorize. Okay. So what are the 4 examples of substrate level phosphorylation? The first one is phosphoglycerate kinase and pyruvate kinase, both of glycolysis. Phosphoglycerate kinase step and pyruvate kinase step of glycolysis. Third one is succinyl thiokinase step of citric acid cycle. The third one is succinyl thiokinase step of citric acid cycle. The last one is creatine kinase of muscle. Okay. So tell me quickly, what are the examples of substrate level phosphorylation? Phosphoglycerate kinase step and pyruvate kinase step of glycolysis, succinyl thiokinase step of citric acid cycle, and creatine kinase of muscle. And other than these 4 examples, every other catabolic step will be an example of only oxidative phosphorylation. So now do you understand why I said kinases? If the name ends with kinases, they are all examples of substrate level phosphorylation. If the name ends with dehydrogenases, they are all examples of oxidative phosphorylation. Because what does a dehydrogenase do? Dehydrogenase removes hydrogen from the substrate, gives that to its coenzyme, which is NAD or FAD, forming NADH or FADH2. Right? That will give rise to ATP. So most of the dehydrogenases are examples of oxidative phosphorylation. Okay. So now do you know the answer for this question, which is an example of substrate level phosphorylation? What will be your answer? Your answer is phosphoglycerate kinase of glycolysis. Okay. So with this, part 1 is over. So part 1 of entire biochemistry revision is over, and I will take this part 2 day after tomorrow, on Tuesday. Tomorrow there will be a session on strategy. Strategies to be followed for NEET PG preparation. So tomorrow at 5 o'clock, that is a session. Day after tomorrow at 5 o'clock, there will be part 2 of entire biochemistry revision. Okay. So thank you all. Get the PDF from the community in Telegram. Okay? No. Community in Unacademy app. My pleasure. I think I will do it as 3 parts, Nairobi. So the next part I will finish lipid and proteins, and the last one will be genetics. My pleasure. Part 2 will complete. No, part 3. 3 parts. Okay. So, see you all.